Well, I would also like to thank ILSI Brasil for the opportunity to present this talk to your group. I want to talk in this first presentation about risk assessment, the doses which might actually provoke allergic reactions in sensitive individuals, and movement towards thresholds by public health agencies. So, food allergies are an emerging public health issue around the world, but they've probably been with us forever. It's just that they were ignored for many, many decades, and now public health agencies are beginning to pay attention, and VISA obviously is as well. And I think I don't have to explain all of the features on this slide. I think the most important reason for the emergence of food allergies as a public health issue is evidence that individuals can die from inadvertent ingestion of allergenic foods. And this was first published in 1988 in the Journal of the American Medical Association by a group led by Dr. John Yoninger, and then again in 1992 by Dr. Hugh Sampson in the New England Journal of Medicine. So the clinical community became very well aware that food allergies can cause deaths, and those early reports have been followed up by many others. But I want to say for the first time in this presentation that fatal reactions require rather large doses. So consumers have become more aware of food allergies. The prevalence of food allergies seems to be increasing in the world for reasons that I do not have time today to explain. But in countries where it's been studied, 4 to 8% of consumers have food allergies. And I don't know what the prevalence is in Brazil because I don't think you have good data. And that data is actually very costly to acquire. And in fact, the United States does not have very good data either. Only the European Union does. And so I think that is a point in the public health issue that could be further emphasized. But as I said, it's going to cost a lot of money. But we know that the reactions can occasionally be fatal. We know that the reactions happen immediately after ingestion of the offending food. We know that the threshold dose for provoking a reaction can be quite low, although the dose for provoking a severe reaction is considerably higher and less well defined because it's unethical to assess. And avoidance is the only strategy for preventing a reaction in the individual who has allergies. So in North America, at least, the United States and Canada, allergens are a leading cause for product recalls from the marketplace. And we also have celiac disease, which is not the same kind of allergic reaction, causes more delayed reactions, but again, a large number of consumers needing to follow gluten avoidance diets. So both allergen and gluten control present the same issues to the food industry. How much is too much? How clean is clean enough when you have shared equipment and shared facilities? Because I think all companies probably do, or certainly most. So I want to focus on a series of questions today. What is the risk to allergic consumers? Do safe thresholds actually exist? And how do these allergy risks get managed in food manufacturing? and then how do the risks communicated to allergic consumers. And I'm going to start with some slides that I took from the World Health Organization. Now, I'm an older guy, but these have been in place since I was very young. So in the early 1980s, this risk assessment paradigm was created by the World Health Organization, and it continues to be used today. There are not very many things that were created in 1980 that are still used today. So this was really wise at the time it was created. And it functions for food allergies as well. So it's a risk assessment or risk analysis process that starts with risk assessment and proceeds through risk management to risk communication. And we're going to focus today primarily on the risk assessment part, because that's the scientific evaluation of the potential health effects from human exposure to allergens in this particular case. But of course, once we do the assessment, we do want to touch upon management issues and communication issues as well. So the risk assessment process itself involves a series of very well-described steps. Hazard identification, what are the potential health effects? Hazard characterization, the evaluation of the nature of the adverse effects, and then whether the degree of intake is likely to be sufficient to provoke a reaction based on what you know about the degree of the hazard. So we have risk management strategies that follow that, because obviously, and these would be included in that guide I heard about this morning, that you must manage the risk once you have identified that it exists. And I don't have time today to talk too much about this aspect, but I do think it's a very critical aspect, but one that's very difficult to accomplish if you don't establish some target level for how much is too much and how clean is clean enough. You must have a target level. If you try to manage to zero, it's hopeless. We can all go home. So, and then communication is critical. These allergic consumers rely upon food labels for preventing their reactions, so we must communicate effectively. When I say we, I mean the food industry. So what is the risk to allergic consumers? Well, that comes from the hazard identification and hazard characterization parts of this risk assessment process, so I'm going to describe those first. In terms of hazard identification, does the food deliberately contain any ingredients derived from a known allergenic source? If it does, you should label them unless they're exempt. And you just had a very good lecture about exemptions. Europe has the best process for exemptions, so I would encourage Brazil to follow that approach. In the United States, we've been less successful with exemptions, and I'll just leave it there. Could the food contain any allergens as a result of cross-contact because of shared equipment or shared facilities, or food chain commingling? And it can be very difficult to assess that final part. And sometime today, I'm going to give you an example of how that has emerged as an issue in the US and Europe. Is food manufactured on shared equipment with formulations that contain an allergenic ingredient? Obviously, in many cases, the answer is yes. But if it is, Do you have effective cleaning strategies? And how do you prove that they're effective? That requires analytical methods, and the analytical methods must be fit for purpose, sufficiently sensitive, but especially sufficiently robust to detect residues in the food that's being analyzed. Is the food manufactured in a shared facility? Well, there could be some risk there too, but I would argue that can be easily addressed with effective allergen controls. So we're back to these key questions. How much is too much? How clean is clean enough? The threshold concept is necessary to answer either of those questions. And unless you have answers to those questions, you may end up with precautionary labels on most of the foods in your marketplace. Ask the Australians. That's exactly what happened in Australia. So do safe threshold levels exist? I'll spare you the suspense. Yes, they do. So we can develop thresholds or reference doses, and they have potential impacts on consumers. So, but we're faced with this entrenched zero threshold concept. And I will submit again, you cannot manage to zero. You can't measure zero, so you'll be paralyzed if you try to use zero. And if you try to use the most sensitive method, be careful, because the most sensitive method may not be the best method. So physicians have always recommended complete avoidance of the allergenic food to their food allergic patients. And so their patients have followed that advice and attempted to practice complete avoidance. Then we have regulators who, because they don't really fully understand the situation, have also adopted laws. The laws in the United States are promulgated by our Congress, which I think has one scientist in the whole body, and they base it on zero threshold, which makes sense to congressional representatives, but makes less sense to scientists. And then the food industry adopts this zero threshold mentality and puts precautionary labeling on almost everything. And then that defeats the whole purpose. But there are benefits to the zero threshold concept. For the public health authorities, this is easy. It's just presence or absence. There's no debate about how much is too much. It makes for a very easy regulatory decision. If you use zero, of course food allergic consumers are protected, and physicians have it easy too, because they can give the same advice to all their patients who are food allergic, complete avoidance. That's what they've been doing for years. And the food industry has limited flexibility with labeling, so they're likely to make less errors. But there are big disadvantages. And I think we heard about those in one of the questions from Vera back in the back of the room this morning. Food allergic consumers will have a diminished quality of life due to limited food choices, because many foods will be labeled or have precautionary labels. The public health authorities are going to spend a lot of time, and in every country they have limited personnel resources. Brazil's not so different in that regard. They're going to spend a lot of time chasing zero instead of chasing the real hazards. Physicians are going to deal with scared and frustrated patients, because if you tell them all that they're the most sensitive individual in the world, they're going to believe that and be frightened. And the food industry is going to focus attention on zero, and then sometimes, as we say in English, miss the forest for the trees, control to zero, but have some big problem that they don't even see. And then I would point out that analytically, zero keeps getting less. You can't measure zero. You have to have a limit of detection for every assay, I would argue that the current assays that we have are sufficiently sensitive, and it's a waste of time and money to create more sensitive methods. So do we have enough information to move away from the zero threshold concept? Yes, I think we do. What will be the impacts of those? There's a lot of them on all the different stakeholders. But what are the impediments to the implementation of some finite threshold concept? And will allergic consumers accept this kind of public health approach? I don't think that last question's easily answered, and would be a big job for Anvisa if they decide to accept reference doses. So we've known for many years that trace amounts of the offending food is sufficient to cause allergic reactions, certainly low milligram amounts. Now I want to pause here and say, I'm going to do a lot of talking about milligrams. That's a dose. Parts per million is a concentration. Milligrams per kilogram is parts per million. You may have very widely accepted foods, but very few consumers are going to eat 1 kilogram at a setting, probably none. But in 2005, a few clinics had started to do very low dose, double-blind, placebo-controlled food challenges, and were able to prove that safe doses existed, and that at low doses, severe reactions did not occur. Many consumers, however, believe that they're extremely sensitive. Many consumers have experienced allergic reactions despite their avoidance diets. And some of these reactions have been quite severe, so the consumers leap to the conclusion that they've suffered severe reactions from low-dose exposures. I would argue that they've probably suffered severe reactions, but from higher dose exposures. But I can't prove that because very few studies have been done quantifying the amounts of allergen eaten by consumers who actually experience a severe reaction. We're trying to put together some of that information now. So in the public health authorities, there seems to be a widespread belief that low doses can provoke severe reactions. That's not true, but it's very difficult to move this concept away. There is also this belief that a zero threshold will protect allergic consumers, because it will. And the fear of setting the wrong threshold seems to overweigh the empathy about the quality of life situation for food allergic consumers. So even if they're willing to accept the threshold information that exists, the authorities want to divide the number by 100 or 1,000 or some other arbitrary factor. And I don't think they need to do that, because we actually have human data on the sensitive subjects in the population. So a few public health authorities have been very brave. The Japanese have established 10 parts per million, that's a concentration, as their regulatory threshold, because they know that the analytical methods that we have today operate effectively at 10 parts per million. They do not operate effectively at parts per billion. So I heard somebody say that they had a food tested to parts per billion. I'd put a big question mark by that one, because I don't know how you do that. So if somebody tells you they can, I'd like to see their evidence. Switzerland established 1,000 parts per million. I don't agree with that, so we'll not mention that again. But I've already indicated how that drives, failure to do something like that drives precautionary labeling. I'm going to skip this slide because it's kind of duplicates what I said on the previous one. So clinical threshold studies have shown that allergic individuals can tolerate the ingestion of small amounts of their offending allergen. And these clinical data can be used to allow the establishment of reference doses, threshold doses, action levels. You can call them whatever you'd like. And consensus is emerging in the United States, I should add Canada, the European Union, and Australia and New Zealand to support the establishment of reference doses. But these have to be discussed with all of the stakeholder groups. That's very important. And you can use statistical dose distribution modeling of the clinical data. So the acronym NOEL means the no observed adverse effect level, the maximum dose that provoked no reaction in a sensitive individual. And the LOEL, the lowest observed adverse effect level, the lowest dose that began to provoke a mild objective reaction in the sensitive individual. We've discovered that it's not good to use subjective doses because they are subjective by definition and variably collected by different clinical investigators. So I don't have time to go all the way back to the original data, but here is an example of a dose distribution. This is the log normal, log logistic, and Weibull distributions of peanut threshold data. So each one of these points represents one or more allergic individuals. And you can see that there's a nice distribution. There are some individuals who are very sensitive, and you'll notice this is a logarithmic scale. There are some individuals who can tolerate more than one peanut in their diet. If you look at their histories, they may have histories of equally severe reactions as the people down here, but it clearly took higher doses to provoke those reactions. So we have that kind of data for a number of allergenic foods, allowing us to begin to answer this question. How much is too much? Low milligram amounts. And that equates to parts per million concentrations. If you drew the line in the sand at 10 parts per million, that would probably be quite protective of that allergic population that exists in Brazil or any other country. So, based on what we know about the threshold doses in peanuts, this amount, 1 milligram, actually I don't have a picture of 0.8 of a milligram. The 1% dose is 0.8 of a milligram or 0.2 of a milligram of peanut protein. That amount is sufficient to provoke a reaction in 1% of the peanut allergic individuals. But 400 milligrams, which is either a half peanut or a whole peanut, depending on which variety you choose, is only sufficient to provoke a reaction in half of peanut allergic individuals. So they're quite variable. So the first time we looked at reference doses was to help the Australians. And the Australian food industry had developed the Allergen Bureau of Australia and New Zealand and developed this VIII program called Voluntary Incidental Trace Allergen Labeling. This is not done by FSANZ, their regulatory public health authority. It's done by a food industry sponsored group. But they established an expert panel. Both Dr. Cravell and I served on it, along with others, including several Australians. And Katie Allen is an allergist, a physician. And we were able to set reference doses for all of these allergenic foods. We had the most robust data from clinical studies on peanut, milk, egg, and hazelnut, but we had sufficient data on soybean, wheat, cashew, mustard, lupin, sesame seed, and shrimp. And we had a lot of data on celery and fish, but the dose distributions were not good, and so we were unable to identify reference doses to foods. I would point out that several of these foods are not on the Australian list: mustard, lupin, and celery. So we also used the European list because we thought there might be broader use for this exercise. So here's the data that we used in 2011. And you can see all of the foods, and you can see the number of subjects. So we had data on 750 peanut allergic individuals subjected to low-dose challenges. We obtained this data from the published clinical literature or from the examination of clinical records shared with us by various physicians in different clinical study centers. And you can see that the number goes down. I think the lowest one that we had where we were able to establish a reference dose was sesame seed with 21. And in most cases, they involve both children and adults. The data set has rapidly expanded. We have data now on more than 1,200 peanut allergic subjects. Sesame seed has increased from 21 to 40. Some of them haven't moved as much as others, but look at the movement of cashew. We have hundreds more data points. This is emerging very quickly. And this is human data from the sensitive segment of the population. And here are the reference doses that we established in 2012. We're in the process of looking at the data with the 2016 data set to determine if we want to change any of these. I don't think any of them are going to change a great deal. You can see that the most potent allergenic food is egg, but that's probably because clinical investigators use raw egg as the challenge material. I would prefer it if they used some kind of processed egg, because I would discourage consumers from eating raw egg in the first place for other reasons besides allergy. Mustard is the second most potent, and peanut is a fairly potent allergen, but not even as potent as milk or hazelnut. I would point out that we now have sufficient data that we can actually establish a reference dose for fish. That will be one of the things that happens in the next round. And here are some of the distributions, just to show you that the allergens are not created equal. Again, egg is the most potent, but they all have these dose distributions. Again, a logarithmic scale. Peanut, milk, and sesame seed are very similar. Hazelnut is somewhat less potent, and soybean is considerably less potent, maybe 100-fold. So we have sufficient data to establish reference doses. And I think reference doses are the only way to bring some order out of the existing chaos generated by the use of zero thresholds. But allergic consumers have to be willing to accept some small level of risk, but its magnitude needs to be explained and clinically demonstrated. And we've just done that with peanut. We did a large study where we provoked allergic individuals, peanut allergic individuals, with the EDO5 dose, the dose that would be predicted to cause reactions in 5% of the individuals. Out of 378 individuals, we saw no severe reactions at the EDO5 dose, and only 8 of those subjects even had objective reactions that met our criteria for inclusion. In my view, public health authorities should use the reference dose concept to control recalls and to limit the enthusiasm of the food industry to use precautionary labeling, and to determine whether the allergen control practices of food manufacturing facilities are sufficient. The industry must use the concept too, again, to control precautionary allergen labeling usage and to evaluate if their controls are sufficient. And of course, analytical method companies have to sell suitable detection methods. And I would argue that the methods on the market now, by and large, are suitably sensitive and specific. So how do allergy risks get managed? Well, with knowledge of the reference doses, food companies can establish corporate target levels. If you don't have those, how are you going to decide what to do? You can use those to guide allergen control measures and labeling decisions. The industry can also assess ingredients to determine if unexpected allergens are present and drive the suppliers to manage allergens more effectively if they find problems. And the industry can use target levels to establish goals for cross-contact control. When you find problems, I would say you don't jump to labeling or precautionary labeling. Risk mitigation should be your first opportunity. You may be able to figure out a better way to control the risk. Now, sometimes those approaches are not economically viable. We can't build separate factories for every different food formulation. That's economically not feasible. But if the exposure dose exceeds the reference doses, then a potential hazard exists and you need to either mitigate it or label it. And so then finally, how are the risks communicated to the allergic consumer? If we have a sensible system, it's really easy. It's called the product label. If it's there in doses that might cause reactions, you have to notify those allergic consumers so that they don't eat the product. I would say that what we had 20 years ago was not good enough because allergic consumers would occasionally be able to find hidden allergens in various food products that would provoke reactions. But what we've had to have today has gone the other direction. Now consumers are over-notified of risks and potential risks through precautionary labeling such that they have very little left to eat in many categories. Thank you.